Hello, everyone. Today, we're diving into a fascinating intersection of developmental biology and microbiology, organoids and microbiome interactions. As budding microbiologists, you've likely explored bacteria, viruses, and fungi in isolation. But in the real world, microbes interact intricately with human tissues. Our traditional cell culture systems fall short in mimicking these complex environments. That's where organoids come in. Think of them as mini organs grown in the lab, 3D, functional, and incredibly insightful. In this lecture, we'll explore how organoids are created, what the microbiome entails, and how these two can be brought together to model real-world biological interactions. We'll look at gut organoids colonized with bacteria, how we can mimic infection in a dish, and how this helps us understand diseases like IBD or gastric cancer. I want this to be interactive, so feel free to raise your hand with questions as we go. Let's begin by unpacking what organoids actually are. Organoids are three-dimensional cell culture systems that closely mimic the structure and function of real organs. They are grown from stem cells, either embryonic stem cells, induced pluripotent stem cells, iPSCs, or adult stem cells harvested from specific tissues. Under the right conditions, these cells self-organize into miniature versions of organs. It's almost like the cells remember what they're supposed to become. For example, if we take stem cells from the intestine and provide them with a matrix like matrigel and the correct signaling molecules, they start forming crypt-like structures, with different cell types such as goblet cells, enterocytes, and Paneth cells, all typical of a human gut. Similar approaches have been used to make brain, lung, liver, and even retinal organoids. What makes organoids so powerful is their ability to recapitulate many key features of real tissues: architecture, function, and sometimes even disease states. For example, organoids derived from cystic fibrosis patients show the same chloride channel defects as the patient's actual tissues. This gives us a living, breathing, sort of model system for research. Let's now talk about what sets organoids apart from other in vitro models. First, they have a 3D structure, unlike traditional flat monolayers. This spatial organization is critical for replicating how cells behave in tissues. Second, they exhibit cellular diversity, meaning they contain multiple cell types found in the parent organ. In the gut, this includes absorptive enterocytes, mucus-producing goblet cells, hormone-secreting enteroendocrine cells, and antimicrobial Paneth cells. Third, organoids are functionally active. Gut organoids can absorb nutrients, secrete mucus, and even exhibit rhythmic contractions similar to peristalsis. And finally, when derived from patients, say through iPSC reprogramming, these organoids are genetically identical to the donor. This opens up avenues for personalized medicine. Imagine testing how a patient's own colon responds to a probiotic or a drug, all without any invasive procedure. Of course, they're not perfect. Organoids typically lack vasculature and immune cells, though co-culture systems and organ-on-chip technologies are addressing that. But for many questions, especially those about epithelial microbe interactions, organoids are a game changer. Now let's shift gears and talk about the microbiome. The human microbiome is the collection of all microorganisms living in and on our body. This includes not just bacteria, but also viruses, archaea, fungi, and even protozoa. The microbiome is most densely populated in the gastrointestinal tract, where we harbor over 100 trillion microbes, 10 times more microbial cells than human cells. These microbes aren't just passive passengers. They digest dietary fibers, produce vitamins like K and B12, and train our immune system. For instance, Bacteroids Thetaeteomicron helps break down complex carbohydrates, while lactobacillus species help maintain gut pH and suppress pathogens. The microbiome's influence extends far beyond the gut. We now know it impacts everything from our brain, through the gut-brain axis, to our metabolism, and even our risk of chronic diseases like obesity, diabetes, and cancer. Dysbiosis, or an imbalanced microbiome, has been linked to conditions like inflammatory bowel disease or IBD, Clostridioides difficile infection, and even autism spectrum disorders. Understanding how these microbes interact with human tissues is essential, but it's also incredibly difficult in vivo. That's where organoids come in handy. Studying the interaction between microbes and human tissues is crucial for understanding how health is maintained and how disease begins. In the gut, for example, the microbiota helps maintain the integrity of the epithelial barrier, modulates the immune system, and competes with pathogens for space and nutrients. When this symbiotic relationship breaks down, we can see the onset of disorders like Crohn's disease, ulcerative colitis, and even colorectal cancer. Let me give you a quick example. In patients with IBD, certain beneficial bacteria like Faecalibacterium prosnitziae are often depleted. This bacterium produces short-chain fatty acids like butyrate, which nourish colonocytes and help keep inflammation in check. When it's missing, you get a compromised barrier and increased inflammatory signaling. Now think about how we could model this. In traditional cell culture, we might use a single epithelial cell line like CACO2, which is flat, lacks diversity, and doesn't behave like a real intestine. Animal models provide complexity, but are time-consuming, costly, and don't always translate well to humans. That's why organoids are so promising. They offer a middle ground, human-derived, multicellular, and amenable to controlled experiments. We can selectively introduce microbes and monitor responses like cytokine release, gene expression changes, and barrier function. Let's now connect the dots. How do we use organoids to study the microbiome? Organoids can be grown in sterile conditions, making them excellent controlled environments for introducing specific microbes. Researchers have used them to study how both commensals and pathogens interact with host tissues. We can examine how bacteria adhere to epithelial surfaces, whether they invade, how they influence gene expression in the host, and what immune responses are triggered. For instance, when gut organoids are exposed to Salmonella typhimurium, researchers observe bacterial invasion, epithelial cell death, and inflammatory cytokine production, just like what happens in the human intestine. This approach offers several advantages. First, it's human-based. No more guessing if a mouse model will reflect human biology. Second, it's flexible. You can use organoids from different patients, including those with specific diseases or genetic mutations. Third, you can precisely control which microbes are present. Want to compare how lactobacillus and E. coli interact with the same tissue? Just add them one by one. Lastly, organoids let us ask questions we couldn't before. How does the gut epithelium respond to an entire fecal microbiota transplant, FMT? How do probiotics alter epithelial function? What happens when we add antibiotics to a microbe-colonized gut model? Organoids open up a whole world of inquiry. Let's zero in on one of the most widely used organoid systems for microbiome research, intestinal organoids, often called enteroids or colonoids depending on the source tissue. These gut organoids are typically derived from adult stem cells residing in the intestinal crypts or from induced pluripotent stem cells. Once cultured under the right conditions, these cells organize themselves into 3D structures that recapitulate key features of the native intestine. They form crypt-like domains and villus-like projections, contain multiple cell types including enterocytes, goblet cells, enteroendocrine cells, and Paneth cells, and show functional polarization. For example, goblet cells within organoids secrete mucins, a key component of the intestinal mucus layer that helps keep microbes at a safe distance. Paneth cells produce antimicrobial peptides like defensins. The architecture supports nutrient absorption and also reflects the compartmentalization seen in vivo, with stem cells residing at the base of crypts and more differentiated cells at the top of villi. Why is this important? Because when studying how microbes interact with the intestinal epithelium, we need a model that mimics this complexity. You can't study mucus interactions in a system that doesn't have mucus. You can't understand pathogen invasion if there's no crypt structure for them to hide in or climb up. Interestingly, organoids even show some functional motility. In some cases, you'll see rhythmic contractions, kind of like peristalsis in vitro. This adds another layer of physiological realism. In short, gut organoids provide a remarkably faithful model to study how intestinal tissues respond to both commensal and pathogenic microbes. In the next slide, we'll look at how scientists actually get microbes into these closed 3D structures, a technical but crucial step for microbiome research. Now that we've explored the amazing capabilities of gut organoids, you might be wondering, How exactly do we get microbes into these organoids to study their interactions? After all, organoids typically form closed spherical structures, like hollow balls, with their apical surface, the side that normally faces the gut lumen, tucked inside. That creates a bit of a logistical challenge. Luckily, researchers have developed several clever approaches to overcome this. Method 1. Microinjection. This is the gold standard for introducing microbes directly into the organoid lumen. It involves using a fine microneedle under a microscope to inject bacteria, viruses, or even complex microbial communities into the center of each organoid. This mimics the natural orientation. Microbes are placed exactly where they'd be in the gut. The downside? It's labor-intensive, technically challenging, and low throughput. But for detailed mechanistic studies, it's extremely valuable. Method 2. Apical out organoids. Scientists have figured out how to reverse the polarity of organoids so that the apical, lumen-facing, side is on the outside. This can be done by disrupting the extracellular matrix and letting the organoids reorient in suspension. Once they flip, you can simply add microbes to the culture medium and they'll interact directly with the epithelial surface. No microinjection needed. This makes experiments much easier and higher throughput. Method 3. Organoid-derived monolayers. In this approach, organoids are dissociated into single cells and then plated onto transwell inserts to form a 2D monolayer with apical and basolateral access. While this sacrifices 3D structure, it allows easy and uniform exposure to microbes and is compatible with co-cultures involving immune cells, barrier integrity assays, and even electrical resistance measurements. Each method has its pros and cons, and the choice often depends on the specific research question. Organoids allow us to track a wide range of host responses in a relatively controlled and high-resolution way. Let's go through a few of the most important responses we commonly observe. 1. Mucus secretion. Goblet cells within gut organoids respond to bacterial colonization by increasing mucin production. This mucus layer is crucial for maintaining a physical barrier between microbes and the epithelial surface. In organoids exposed to commensal bacteria, we often see a thickening of this layer, a sign of a healthy defensive response. 2. Antimicrobial peptide production. Paneth cells and enterocytes can release antimicrobial peptides such as defensins and lysozyme. These peptides help shape microbial communities by selectively inhibiting certain bacterial strains. For example, organoids exposed to Salmonella enterica show a marked increase in antimicrobial peptide secretion. This is part of the innate immune response. 3. Cytokine signaling. One of the most accessible readouts is the expression and release of pro-inflammatory cytokines. IL-8 is commonly used as a marker. It attracts neutrophils to the site of infection. When organoids are colonized by pathogenic strains, you often see a spike in IL-8 levels, whereas commensals tend to induce a much milder cytokine response. 4. Barrier integrity. Organoids can also be used to study how microbes affect epithelial tight junctions. Pathogens like enteropathogenic E. coli can disrupt these junctions, leading to increased permeability, a hallmark of gut disorders like IBD and leaky gut syndrome. Researchers often assess this using fluorescent permeability assays or by tracking changes in tight junction protein expression. 5. Cell proliferation and death. Some microbes promote epithelial regeneration, while others induce apoptosis. For instance, Helicobacter pylori can trigger cellular proliferation, a mechanism thought to contribute to gastric cancer development. Let's walk through a compelling example of how organoids are being used to dissect host-microbe interactions, the case of Escherichia coli in human gut organoids. Now, we all know E. coli is a diverse species. Some strains are friendly, part of our normal microbiota, while others are notorious pathogens like EHEC, enterohemorrhagic E. coli, or EPC. Using organoid models, researchers can investigate how these different strains behave in the same human epithelial context. In one landmark study, scientists microinjected 2 different strains of E. coli into human collonoids, one commensal, the other pathogenic. They found that while both colonized the lumen, only the pathogenic strain triggered a robust inflammatory response. IL-8 secretion was upregulated several-fold, and there was visible disruption of tight junction proteins, leading to increased epithelial permeability. Interestingly, the commensal strain didn't just behave passively. It actually stimulated low-level mucin production and AMP release, suggesting a mutually beneficial relationship that helps maintain barrier integrity and immune tone. Another key observation: pathogenic E. coli altered the balance of cell proliferation and death within the organoid. They caused apoptosis in some enterocytes and stimulated overproliferation in others, a response that might underlie tissue damage or even precancerous changes in chronic infections. This kind of detailed, cell-type-specific insight would be nearly impossible to extract from in vivo models or 2D cell lines alone. It shows the power of organoids not just to model infection, but to capture subtle, dynamic interactions between microbes and human tissue in real time. So, as you can see, organoids are not just lab curiosities. They are functional mini-organs that help us recreate and study complex biological phenomena in a dish. Let's now turn our attention to another fascinating and clinically important case: the interaction between H. pylori and gastric organoids. H. pylori is a spiral-shaped, gram-negative bacterium that colonizes the stomach and is a major risk factor for gastritis, peptic ulcers, and gastric cancer. Over half of the global population carries H. pylori, but only a subset develop disease, raising an important question: what host factors determine susceptibility? This is where gastric organoids come into play. Researchers have cultured organoids from human gastric tissue and exposed them to H. pylori, either by microinjection into the lumen or by modifying the organoid polarity to allow apical exposure. The results are quite telling. Upon colonization, H. pylori activates a well-characterized virulence pathway involving a protein called Kage, which is injected into host cells via a type IV secretion system. In organoids, this interaction leads to striking cellular changes: elongation of epithelial cells, disruption of tight junctions, and activation of signaling pathways which are associated with inflammation and even oncogenic transformation. One of the most important findings was that H. pylori colonization led to increased expression of IL-8 and other pro-inflammatory cytokines. These responses mimic what we see in early stages of H. pylori-associated gastritis, showing that organoids can effectively model disease initiation. What's even more powerful is the ability to use patient-derived organoids. For instance, organoids grown from individuals with known polymorphisms in inflammation-related genes show exaggerated responses to H. pylori exposure. This gives us a platform to study host genetic susceptibility in a controlled setting. In short, organoid models of H. pylori infection provide critical insight into both the pathogen's virulence mechanisms and the host's molecular response, offering a window into the early events of gastric disease and cancer. So far, we've been discussing epithelial organoids in isolation. But of course, in vivo, the gut isn't just a lining of epithelial cells. It's an immunologically active site with constant crosstalk between epithelial cells, immune cells, and microbes. So the next frontier in organoid modeling is co-culture, particularly with components of the immune system. Several research groups have begun to combine organoids with immune cells such as dendritic cells, macrophages, T cells, and ILCs. This allows us to study how the immune system responds to microbial colonization in real time and how epithelial cells, in turn, signal back to immune cells. Let me give you a concrete example. In one study, researchers exposed intestinal organoids to Salmonella typhimurium, then co-cultured them with human monocyte-derived dendritic cells. The dendritic cells were able to sense microbial signals indirectly through cytokines secreted by the epithelium and began upregulating maturation markers and antigen presentation machinery, much like what we see in a normal mucosal immune response. In another model, researchers introduced IL-22-producing ILCs into colonoid cultures. When bacteria were introduced, the ILCs responded by stimulating the epithelium to produce antimicrobial peptides and repair factors, recapitulating the protective effects seen in vivo. These co-culture models allow for sophisticated experiments. For instance, by using immune cells from patients with inflammatory diseases like ulcerative colitis, you can test whether their immune responses to bacterial exposure differ from healthy controls, and perhaps even screen for immunomodulatory drugs. It's still an evolving field, and challenges remain, such as maintaining immune cell viability and function over time in culture, but the potential is enormous. In summary, integrating immune cells into organoid systems brings us one step closer to modeling the full complexity of the mucosal immune microbiome interface. As we move into the future of this field, several exciting trends are emerging that promise to elevate organoid microbiome research even further. First, we're seeing a surge in multi-omics integration, that is, combining transcriptomics, proteomics, metabolomics, and epigenomics to get a comprehensive picture of how organoids respond to microbial signals. For example, researchers have exposed gut organoids to defined microbial consortia and then performed RNA-seq to map changes in gene expression, followed by mass spectrometry to detect shifts in secreted metabolites and proteins. This systems-level view is critical. It helps us understand not just isolated pathways, but the net effect of microbial colonization on host physiology, from metabolic rewiring to changes in chromatin accessibility. Second, there's a growing emphasis on personalized organoid models. Because organoids can be derived from individual patients, they capture unique genetic, epigenetic, and disease-associated features. For example, collonoids from Crohn's disease patients may show exaggerated cytokine responses to the same bacteria that elicit only mild effects in healthy organoids. This opens the door to precision microbiome therapeutics, where we could, one day, match a patient with a specific probiotic, prebiotic, or microbial metabolite tailored to their biology. Third, researchers are developing synthetic microbial communities, simplified, defined consortia that mimic key aspects of the native microbiota. By introducing these communities into organoids, we can map how specific bacterial taxa interact with the host, with far more control than in complex fecal samples. And finally, organoid-on-chip systems are pushing the boundaries by incorporating microfluidics and mechanical forces. These allow for dynamic flow, gradient formation, and even peristalsis-like movement, all of which influence how microbes colonize and interact with epithelial surfaces. As promising as organoid-based microbiome research is, we need to acknowledge the challenges. First, one of the main limitations is the lack of full immune complexity. While we can co-culture organoids with immune cells, they don't yet fully replicate the complexity of the GALT or systemic immune interactions. The signaling cascades, antigen presentation, and feedback loops of a whole immune system can't yet be modeled in a dish. Second, vascularization and oxygen gradients are missing in traditional organoid systems. Many gut microbes are obligate anaerobes. This makes it difficult to co-culture them with epithelial cells unless special conditions are created. Microfluidic organoid-on-chip systems offer some solutions here, but they're still under active development. Third, there's a lack of long-term stability and reproducibility. Organoids derived from different patients, or even from the same person at different times, can behave differently. That's a strength for personalized medicine, but a challenge when you want consistent, reproducible results across experiments. Fourth, most current systems are limited to single tissue models. But in real life, organs interact. Some labs are now working on multi-organoid systems, where gut, liver, and brain organoids are connected on a microfluidic chip. This could be a game changer for studying the gut-liver axis in diseases like NEFLD or the gut-brain axis in conditions like Parkinson's. Lastly, microbial diversity is hard to preserve. Many studies rely on simplified microbial communities or even single strains. While that's scientifically useful, it doesn't always reflect the rich, competitive, and cooperative microbial ecology of the human body. Despite these challenges, the field is evolving rapidly. Technological innovations, such as CRISPR-engineered organoids, anaerobic co-culture chambers, and improved organoid differentiation protocols, are helping to overcome many of these hurdles. To wrap things up, let's quickly recap what we've covered and why this topic matters. We started by discussing organoids' 3D, self-organizing structures that model real human organs in vitro. They allow us to recapitulate tissue structure, cell diversity, and function in a way that was unimaginable just a decade ago. Then we explored the human microbiome, the complex, dynamic community of microorganisms that live on and within us. The core of our lecture focused on how organoids provide a transformative platform to study microbiome-host interactions. Using gut organoids, researchers have been able to model how commensal and pathogenic microbes interact with human tissues, investigate immune responses, study personalized responses to microbes using patient-derived models, and incorporate immune cells to simulate inflammation and track long-term effects. We looked at real-world examples like E. coli in gut organoids and H. pylori in gastric organoids, which illustrate how this research can uncover mechanisms of disease and point toward new therapies. And we acknowledged current challenges: the absence of full immune systems, anaerobic microbial constraints, limited vascularization, and reproducibility issues. But despite those, the field is moving fast toward more complex, integrated models that will redefine how we study host-microbe interactions in health and disease. So if you're a student thinking about where microbiology is headed, this is a field brimming with opportunity. Whether your interests lie in infectious disease, cancer biology, immunology, or even bioengineering. My takeaway for you today is simple. Organoids are not just models, they are windows into the complexity of human biology. And when we pair them with the microbiome, we gain powerful tools to understand ourselves, our diseases, and our potential treatments like never before. Thanks for your attention.